Hello, everyone, and welcome to another day of the second wave of the pandemic, science and society. Rose, is my sound coming through okay? Great. Okay, perfect. Thank you. So we're going to go ahead and get started with today's lecture. Okay, great. So today we're going to be talking about mutations and selection, and there is a wide variety of different students in this class from different backgrounds. So I am going to review some basic information at the beginning so that we're all on the same page. Some of you might know information about mutation and selection really well. Others of you might not. So we'll just go over some basics, and then that'll be integrated into what we're talking about today in terms of COVID and SARS-CoV-2 mutations. So I want to start with a poll, actually. We have these two pictures on the screen here, and these pictures represent a very classic example in science of a particular phenomenon. And I am wondering how many of you, given the different backgrounds that all of you are from, how many of you are familiar with this example? So I want to do a poll, which we're going to start in a minute, where you select what you think these two pictures that are on the screen, what you think these two pictures are representing, what this scientific idea is related to. Jen, could you share the poll now? Thank you. Okay, so go ahead and take a minute to look over this question. What is this an example of, these two pictures? And choose the answer that you think this is related to. Just a few more of you still need to vote, and then we'll talk about it. All right, we have about 94% of the vote in. Hopefully the rest of you have had a chance to look at these two pictures. So we're asking, what are these pictures an example of? Okay, so I'm going to go ahead and end the poll. All right, so 79% of you chose evolution, which is correct. Some of you chose zoonotic disease transmission, some of you chose disease vectors, and some of you chose land conversion to agriculture. So we have, a range of backgrounds in this class. So let's talk through this example a little bit. All right, so, and I see someone has put in the question and answers, it's peppered moths and the Industrial Revolution. Very good. So here we have an example of evolution. So this is a classic example that's often given when people are learning about evolution. On the left of the screen, this is a peppered moth that's on a tree in England prior to a lot of industry coming into England. And so the bark of the tree is white, and you can see that this moth is white, and it blends in really well to this background of the tree. Now, this is important because it prevents the moth from easily being seen by predators. And so it's less likely that the moth will be eaten because it's blending into the background so well. So this is a good fit when you think in terms of a trait that an animal could have and the environment in which it lives. This is a good fit, right? And so you have a lot of these lightly colored or speckled peppered moths. Then we have the Industrial Revolution, in which what we breathe now commonly starts being spewed out into the air. And the background now becomes very dark because instead of being this white birch tree, you have soot and other contaminants in the air that land on the trees and create this dark colored, basically a dirty bark on the tree that now you can see on the left side, this white moth stands out against this dark background. Hopefully you can also tell that there is also a black moth next to the white moth on that dark background. Now, originally, white moths were very common because, again, they had this protection from predators. However, once this pollution started changing the color of the trees that the moths were spending time on, the white moths stood out more. They were more easily seen by predators, and therefore they got eaten more. Now that has implications for the changes that we see in this population of moths. And I want to walk through some of those as well as a few other examples before we start talking about COVID. So one thing is that you should be aware that there is variation in all populations. So the variation that exists in humans, in other animals, in these moths, there's always variation within a population. I'm sure by now you are all aware that we are made up of different genes. What combination of genes we have varies between individuals. So in the case of the moths, there was always variation in color of the moth. That existed, but which color was more beneficial given the environment varied when there was this change in industrial production. And because there's variation, there's differential survival and reproduction. So different individuals in a given environment are better at surviving and reproducing. In the example on the right, this light-colored moth would be well fit to this environment and be able to survive and reproduce in that environment. Whereas, sorry, that's on the left. On the right, this white-colored moth stands out in this environment, so it is less likely to do well at surviving and reproducing. That, my friends, is evolution. So evolution is simply a change in allele frequencies of a population over generations. People often think of evolution as what we call macroevolution, where you actually get speciation and you see different species resulting from evolution. But in fact, before any sort of speciation happens, evolution is a process in which there is a change in allele frequencies of a population over generations. So let's break that down a little bit. Some of you may not be familiar with the term allele. An allele is a variant of a gene. So as I said before, we have a bunch of different genes that make up the different physical traits that exist in us and some behavioral traits as well. Alleles are gene variants, right? So they're the different variants that you could have for a given gene. So for example, with the moths, their gene for what color they are, which likely is actually multiple genes, but they could have different variants so that they either are a lighter color or a darker color. Okay, that's an allele. So in evolution, what we see is a change in the alleles. Maybe at first there were a lot of alleles that made white moths. And then when you see this change in the environment, there is a shift in the next generation to being more of the alleles that code for dark coloration in the moths. So we want to look at how these alleles frequencies change in a given population, like for example, the moths in England, over generations. The reason it's over generations is because these changes can only happen when reproduction happens. The changes aren't happening within living organisms because they have a set of alleles, but then which ones become more prominent in the next generation can change. Okay, so within evolution, there are 4 forces of evolution that we realize lead to these changes in allele frequencies. That includes mutation, genetic drift, gene flow, and natural selection. Some of you will be very familiar with this, but again, I want to make sure that we are all on the same page here. So for today's lecture, we're really interested in 2 of these forces of evolution, mutation and natural selection. So I'm going to talk specifically about mutation and natural selection, and then we'll start wrapping them into our understanding of the different mutations and variants of SARS-CoV-2. Okay, so mutation is a random change in the genetic code. Mutations are important because they're the ultimate source of novel genetic variation. Novel just means new. So these mutations are a way to introduce new variation into a population. And remember that that variation is very important because that's what evolution is able to take advantage of when there are changes in the environment. Mutations are relatively common, but most mutations are neutral or nearly neutral. So that means they really don't have an effect on the individual. Some are deleterious, which means that they are bad for the individual. If you have a mutation that isn't helping your survival and reproduction, it likely won't last for long in the population because it doesn't confer an advantage. Instead, it confers a disadvantage, and it's less likely that that individual will be able to compete to survive and reproduce. But sometimes you have mutations that are beneficial to an organism. It makes them better at surviving and reproducing. Now, this sounds like a very positive thing, right? But when we're talking about a mutation in a virus that makes it better at surviving and reproducing, that actually accounts for something really problematic for us in terms of that mutation. Okay. Then we have natural selection. So mutations have introduced new variation into the gene pool, and then natural selection can act on that variation. Natural selection is the process by which certain traits of an organism enable it to survive and reproduce in an environment, thus increasing the frequency of those traits in future generations. So again, this is giving it an advantage in terms of survival and reproduction within a given environment. And that means that that trait or those alleles will increase in frequency in future generations. And this is the idea of differential survival and reproduction. So here I just have a picture that I took of, two lizards, a bigger lizard eating a smaller lizard. This idea, predator-prey relationships, those sorts of things are easy to think about when we think about evolution, mutation, and natural selection. So this idea of mutation and natural selection has been shown in a lot of different organisms. These are things that we can measure in the span of our lifetime, looking at changes in allele frequencies in populations over generations. A similar example to the peppered moth example is looking at mice on volcanic rock. There are studies of these mice in which the mice that are living not on volcanic rock have this white coloration, and then populations that moved into areas where there was volcanic rock and were living on volcanic rock stood out very easily because the white color of their fur made them very easy to be seen by predators, which are hawks and things like that flying overhead. And so there was a mutation that resulted in a change in coloration for some of the mice. Now that mutation, which caused their fur to turn to a darker color, was so advantageous that in relatively few generations, most of the mice living on the volcanic rock had this darker coloration. And again, it was the combination of a mutation and the advantage that that mutation conferred that led to this change in the population. Now, we've known about evolution and this process of selection for a long time. And in fact, it's part of the reason that we have so many different dog breeds, for example, or if you grew up on a farm, you know that artificial selection is also used in farming. So there can be sources in the environment that select for certain traits. For example, in a giraffe pictured here, the need to access food in certain habitats led to this long neck in giraffes. Whereas in dogs, it's not that we have all the different types of dogs that we have because there was some natural situation that resulted in dog variation, but rather that's artificial selection, people taking what we know about how this works in the wild and applying it to things that we want to see changed in our own lives. And so selectively deciding a certain variation of a trait in a given individual is something that we want to see more of, and then selectively breeding those individuals that possess that trait. That's how you end up with so many different dog breeds, and breeds that are selected for certain things, right? Like breeds that are good in the water, breeds that are good at hunting, breeds that are good with children. My dog that's a pit bull mix, good at spinning around in circles and cuddling a lot. There's all these different traits that can be selected for. And artificial selection is just people doing that selecting that normally happens in nature because of the environment and the different pressures of the environment. Okay, so we now know that the example of the moths in England was because of a mutation. So there was a mutation that led to this variation in which you have some dark colored moths and some light colored moths. And similar to what we started with, this light colored bark prior to the Industrial Revolution was a better match for the lighter colored moths. And then after the Industrial Revolution, when there were darker trees, the white moths stood out, and there was a benefit to the darker colored moths. So when they graph this over time, you can see here that the number of white moths, which are these white circles, was higher originally compared to very few dark moths in these black squares. But then over time, after the Industrial Revolution, you have this very clear selective pressure. That is the ability to hide from predators, right? And so pretty quickly, you see this drastic shift in which the number of white moths go down quickly. So, once you're eaten by a predator, you can't reproduce anymore. So naturally, if you are standing out and easy to be seen by a predator, you get eaten, you don't produce any more offspring to pass your genes on to future generations. And so it makes sense then that you would see this very sharp decline in the white moths in response to that change in the environment. And then what that means is that the black moths had less competition. They were able to hide easily from the predators. And so they had this very steep increase in numbers. So that's what we're talking about when we're talking about mutation and natural selection. Okay. Now, we all know that this is a concern in health fields, and it's very important for people in health fields to understand evolution because these same principles of mutation and natural selection can impact our health. One of the clearest examples that we have in modern medicine is antibiotic-resistant bacteria. So this figure shows you, for example, how when you have a normal, random population of bacteria, there will be some resistant bacteria in here. And remember, that's because variation exists in every population, right? So you have this variation within this population of bacteria, but then these antibiotics act as a selective force. The antibiotics are a selective force that are killing off all of these bacteria that are susceptible to antibiotics. And the only ones that are surviving are very few, including some resistant bacteria. Those resistant bacteria then have an advantage because they don't get impacted by these antibiotics. And so they then can reproduce in much greater numbers. And then what your resulting population is, several generations later, you have this resulting population that's mainly resistant bacteria. So it's important for us to understand how evolution works when we're thinking about different threats to our health. Yes, okay. So I want you to be thinking about this example, and I'm going to come back to it in terms of vaccines and COVID-19. I don't think there are any questions about this example. I'm double checking, but I just want to make sure. Yeah, I mean, you know, the reproduction time of different species, even different species of bacteria vary, but in bacteria, you know, there's really quick reproduction, which is one of the reasons that we can see this in hospital settings, for example. You know, there's been a lot of work in recent years on antibiotic resistant bacteria and the concerns about that within our hospital settings. Okay. So when you think about COVID-19 and SARS-CoV-2, I want you to remember that every transmission event, every opportunity that the virus has to replicate is an opportunity for mutation. So this act of reproduction is important. That's a critical point in mutation, right? So every time we decide to let down our guard and, you know, go to the Super Bowl or go have Thanksgiving meal with a big group of people who all came in from out of state and whatnot, all of these events then open up the opportunity for mutation to occur. So given that every replication of the virus is an opportunity for mutation, it's a good argument against letting the virus run rampant and not enforcing or practicing prevention measures, right? So we've talked about some places where they tried, you know, they thought from the beginning, let's just get to herd immunity. Let's not even try and stop what's about to happen. And, you know, in the United States, we've had a fuzzy kind of that version of practice in some places. But if we are concerned about mutations, then this is actually not a good way to try and deal with COVID-19. Okay. So we now know that there are different strains of the virus circulating. And I see people asking questions about, like, you know, can the virus transmit more easily and so on and so forth. That's what this entire lecture is about. So just hang tight in terms of asking your questions. So we know that there are different strains of the virus circulating. And we also know that there have been different strains circulating for the majority of this pandemic. The very beginning of this lecture was all about how variation exists in every population. So there has always been variation in SARS-CoV-2. There's always variation in every population of a virus, of animals, and so on. So that isn't what's concerning at this point. The ability for certain different variants to then become more common and pose more serious concerns, that's what's concerning to everyone. So we've had different strains circulating for the majority of the pandemic, but now there are particular strains or variants that are of concern. So I'm going to walk us through those. So the first one I want to mention is from the UK. And you may have heard of this one. It's one that's being talked about in the news more than others. So VOC stands for variant of concern. And this variant of concern, it's called B117, has higher transmissibility than the non-variant of concern lineages. So in other words, all of what we were mostly dealing with for last year, the SARS-CoV-2 strains that were infecting most people around the globe, are less transmittable than these new variants of concern. This B117 variant of concern was first detected in November 2020 in England, and it likely emerged, they believe, in September 2020 in London or somewhere near London. So it's been circulating for about 5 months maybe now. We know that there is an increased binding and infectivity compared to the original strain that we were dealing with. So that means that the virus is better at binding to our cells and infecting our cells. You'll remember that coronaviruses are known for those spike proteins. So a lot of what you're going to be hearing about in terms of different variants is changes in those spike proteins that make it easier for the virus to bind to our cells and also infect our cells. And that is obviously a reason for concern. We also know that this B117 variant potentially could make some testing less effective because the tests, PCR testing in particular, is looking for certain genetic markers. So when you have genetic changes, which is what a mutation is, it's a change in the genetic code. When you have changes to the genes, to the genetic code, that can complicate some testing. To what extent the testing, different types of testing is still effective or not against in detecting these different strains, I think is still somewhat unclear. But certainly it appears that most of these variants that I'll talk about today are similar enough to the original strain of SARS-CoV-2. These are all, by the way, SARS-CoV-2. They are different strains of the same virus. So to what extent does this complicate testing? I don't think we know that entirely yet. And you'll hear me say that a lot because we still don't know a lot about these things. You know, this is very new. As of November, people were aware of this. And this you'll see in the upcoming slides that November is actually the furthest out of any of the variants of concern that we know about. So this is all really new information. Okay, so this B117 variant has 17 mutations. So the mutations that have been identified that vary in this strain compared to the original SARS-CoV-2 that people were studying, that genome that was sequenced, there are 17 mutations. 8 of these mutations change the shape of the coronavirus spike, which as I mentioned before, that's what the virus is using to attach to cells. So these change in shapes of the spike is really important. One mutation in particular makes the virus more contagious. So it's not only about what's happening once it's in the body, but also in being able to be transmitted. 2 of the mutations make it harder for antibodies to attach. So our body's defense system, which relies on antibodies to identify foreign bodies in our system, 2 of the mutations in the B117 variant make it harder for antibodies to attach. Studies that have been done so far suggest that this variant is 50% more transmissible than the original strain that was circulating in the UK. So this is one of several new variants of concern. Again, this is the one that was detected most earliest on in December of last year. So you can see here, it's SARS-CoV-2 lineage B117, but it's also called variant of concern 2020-1201. And this you'll notice on some of the other variants of concern, this is referencing the dates, right? So this one was designated in 2020 in December. So keep these numbers in mind as we're looking at them, because what you'll see is that some of these new strains were designated in 2021 and we're only in, we're not even to mid-February 2021 yet. So what we're talking about are things that are very, very new and that we still need to learn a lot about. Okay, so the second variant of concern that has been identified is from South Africa, and it's 501YV2. This one has 8 mutations, and of those mutations, some of them help also with attaching to human cells. So again, we have these changes that are related to how these variants are binding to human cells, and that has obviously implications for human health. So I think, again, the one that we know the most about is the B117 variant, because it's been a few months since it was identified, which still in the grand scheme of things is not very long. I'm going to talk to you more about some concerns about the South African variant, because there are concerns particularly around vaccines. But for now, I'll say that, again, this is something that we are still trying to understand more about. And so aside from some information about the biology and some preliminary tests, looking at how it interacts with vaccines, for example, we don't have a lot of information still. Though both the B117 variant and this 501YV2 variant are more contagious. So they're measuring, I see some questions about how do you know they're more contagious? So they're measuring this by looking at, retroactively looking back at spread that's occurring, and looking at how many people were infected with one strain or another. And then from that, how many more people became infected. And with that, they can see this very quick transmission of these new strains. Okay, as I mentioned, people are asking questions about, will the vaccine be effective and so on and so forth. I'm getting to that, so just hold tight for a little bit. Okay, so are these variants more deadly? Well, as I said, I'll say a lot this lecture. It's really too early to tell. We don't have a lot of information yet on how deadly these different variants are. However, potentially, yes. The one that we know the most about, as I've said already, is the B117 variant in the UK. And there was a paper presented at the SAGE meeting, so associated with the World Health Organization, that says there is a realistic possibility that variant of concern B117 is associated with an increased risk of death compared to non-variant of concern viruses. The way that they determined this was by looking at the people infected with these different variants and what the outcomes were for them. So this particular paper, which you can find online if you're interested, outlines some of the different methods that they looked at to compare. And there was, a range of like basically 1.35 to 1.91 times the concern for this new variant compared to the original strain. So, although we still need much more data to know how deadly it is and how concerning it is, there is some evidence that potentially it could be more deadly. Okay, the good news here is that the vaccines being used in the US appear to be effective against these known variants, or at least somewhat. I am going to walk us through this information. So I first want to note information about the vaccines. Now, please keep in mind that we are going to have a specific lecture on vaccines, including that we will get to talk to the Deputy Director of the FDA, who is responsible for vaccine development and distribution. So I'm not going to go into all the specifics about the vaccines, and I'm saying this now for those of you who are listening to please not ask a bunch of questions about those specifics, because that's something we'll talk about at a later point, and I want to make sure that we can get to this information about different strains. But I think it is worth us knowing, as we think about the potential for the vaccines to work with these different strains, that there are currently 2 vaccines that are approved by the FDA for use in the United States. They both use a similar technology or a similar science, in that they are both mRNA vaccines. And actually they're very, very similar. So these 2 vaccines are extremely similar. We can expect them to be responding to different strains, different variants of concerns in a similar way. So those 2 vaccines, the first one was produced by Pfizer BioNTech, and the other one was produced by Moderna. So I'm sure that you've heard those names. These are the 2 vaccines that are available within the United States. You may have also heard about a Johnson & Johnson vaccine, and I know that a lot of people have questions about the Johnson and Johnson vaccine. But one thing that I want you to be aware of is that the Johnson & Johnson vaccine has not been approved by the FDA yet. That doesn't mean that it won't be, potentially it will, but what we have information about right now are the two vaccines that are being used in the US. Also, the Johnson & Johnson vaccine uses a different type of technology than these other two. So what I say about these new variants and vaccines will be relevant for any mRNA vaccines, not necessarily for every type of vaccine. The other thing that I think it's important for you to keep in mind is that other countries have also developed other types of COVID vaccines. So there's different types of technology being used in different countries. China has developed vaccines, other places have developed vaccines. So what I am talking to you about today in terms of how these different variants, what we know about the different variants and the vaccines that are available is very much based on the US vaccine distribution, which are these two mRNA vaccines from Moderna and Pfizer. Okay, so as I mentioned already, we will have a lecture on vaccines later in the semester. So I'm not going to focus on the specifics of how the vaccines work, but I am going to address concerns about how the different SARS-CoV-2 variants will work with the two FDA-approved vaccines in the US. So those Moderna and Pfizer vaccines. Okay, so the good news is, as I said, the vaccines being used in the US appear to be effective against the known variants, or at least somewhat, and I'll walk you through that. So one thing that I want to say at the beginning, and I'll say it again at the end of this bit of lecture, is that as this vaccine rollout is happening, we should continue to take precautions to prevent variants that are not controlled by the vaccines that have been developed. I mean, the vaccines themselves are 90 to 95% effective. And so we should be taking precautions as this rollout is happening, particularly as many people, including more vulnerable populations, are not given access to the vaccine right away. So I'm going to continue to urge you, please, to try and do what you can to reduce the transmission. So I want to go back to this image that existed before, and you'll notice that with my excellent PowerPoint skills, I've created blue boxes over the words bacteria and stuck in the word virus, all of these places. And then I covered up antibiotics and I put in vaccine. I did not actually think to create a little shot figure here instead of pills, but you get the point. So similarly, what we're worried about when we're talking about mutations in selection related to the COVID-19 vaccine is if we may see a similar situation where there is this selective pressure, the vaccine, that maybe deals very well with some variants of the virus, but then results in a situation where more difficult to deal with variants of concern, are able to replicate much more. Now, keep in mind, in terms of a virus, in order for this replication to happen, there needs to be people that it's replicating in, right? There have to be people that are transmitting the virus and that the virus is able to replicate in. So I think that a lot of people have been waiting for the vaccine as the savior that's going to take care of everything for us. But the reality is that was never going to be the case. And if you took the first pandemic course, you're used to me saying that. We really have to implement certain safety behaviors so that we can deal with this virus overall. In addition to using vaccines, there is some personal behaviors that have to be used and policies at a bigger level. Okay, so some of you may be familiar with Dr. Corbett, who was one of the people that developed the Moderna vaccine. And she's been featured in a lot of news programs, which may be why you've you've seen her or heard of her. So this is someone who developed the vaccine, has been a great spokesperson for trying to get people to get the vaccine. She's done a lot of excellent work in terms of talking about how the vaccine works, people's concern about the vaccine, a lot of great health messaging around vaccines. So this is someone who obviously has a vested interest in seeing the success of that vaccine. And even she is warning us all how important it is that we take precautions as the vaccine is being rolled out. And she says here in this social media post from the beginning of the year, viruses need hosts to replicate and mutate. And she says, I feel like we need a "You Too Can Prevent New Mutations" campaign. Where is Smokey the Bear? So trying to get out this message that our behaviors play a role in how successful these vaccines are. And as I've said before in this class, we've had this incredible scientific feat of getting the genome sequenced very early, getting that genome sequence to people who could then incorporate it into the technology they were working on vaccines already, and create very quickly this these vaccines that we're using. It's incredible. So now we need to do our part also to make sure that the vaccines that are being distributed are distributed and not, that effort isn't being taken advantage of or disturbed by people just not doing what they need to do to try and stop this pandemic. Okay. So there's a question about how many people need to be vaccinated in order for us to have herd immunity. The number on this keeps changing a little bit. And again, I think that, the time that we talk about vaccines will be a better time to ask that question. But roughly, I think that people are suggesting that 75 to 80% of people vaccinate. So 75 to 80% of the population needs to be vaccinated for us to be at a level where we'll be able to get back to some version of normal, right? And that's why I keep saying to all of you, the vaccine rollout is going to take the better part of this whole year, so get ready for it. And the more we do to prevent transmission, actually, the more successful that rollout will be and the better off we'll all be. So, I'm going to talk a little bit more about that later on, but I definitely think that similar to what we talked about in terms of Sweden, for example, like not controlling the virus early on actually just leads to more and more problems. In the same way, not controlling things right now as we're rolling out these vaccines will be more of a headache later on. If we can try and really do our part right now to whatever extent that we can, that will be better in the long run. Things will go back to normal, quote unquote, faster if we can do that. Okay. I am going to talk about reinfection. I see some of you have questions about that, so just hang tight. Okay, so I've talked to you about the UK variant. I've talked to you about the South African variant. Most recently, so this is saying early December because that's when it started circulating, but you can see here in the variant of concern number that this is really something that was designated in this year in 2021. The most recent variant that has become a concern that we have started to hear about is this P1 variant in Brazil. So this variant is circulating in Manaus, Brazil, which is in the Amazon. So you may have heard, maybe you haven't, but this is a massive city. It's a very big city with lots of people, and it was talked about a lot last year because actually there was a horrible spread of COVID in Manaus last year. That resulted in 75% of the population being infected with COVID last year. So in terms of thinking about how much do we have to worry about these new variants if we have antibodies because of a vaccine, or if we have antibodies because we already had COVID. The information coming out of Brazil is very concerning because in Manaus, 75% of the population was infected with SARS-CoV-2 last year. then they now are going through this second massive infection with people who have already had COVID getting it again as part of this new strain. That obviously is very concerning. So for example, there is a paper that shows a confirmed reinfection. This is the paper that's available online. And it's looking, taking one patient, for example, showing their confirmed COVID infection from previously with the previous strain, and then now showing a new infection with the new strain, and actually having the genetic data to show that. So, this is just one example, but I think when you think about a city in which 75% of the population was infected with COVID-19, you think about a city that has basically reached herd immunity, and you think like, okay, then they're going to be okay moving forward. And the really concerning thing about this new variant is that it gets into a city like that and then just reinfects everybody all over again. And that is very concerning. So some people are asking about the time frame between the reinfection. Was it greater than 3 months? I'm not sure why 3 months specifically is being asked. But yeah, this is one of the questions originally, and this is why I show this particular citation about someone being reinfected. This is one of the questions originally. When people started getting reinfected in Manaus and they weren't sure what was going on, the big question was, is this just showing us how you can get reinfected with SARS-CoV-2 over and over again? Like, if enough time passes, can you get reinfected? So that is still a question. That's still a very important question. And I know a lot of you have questions about, are we going to have a COVID vaccine every year like we do the flu shot? Again, let's ask that question during the vaccine day. But I think in terms of what's happening in Manaus, the concern is, originally the concern was, is this just us seeing that after a time period, people can start to get reinfected? And while that may be the case, I think that some of the data are showing in particular that it is not people being reinfected with the same strain, but instead people being reinfected with this new strain, which is, I mean, either way it's concerning, right? And either way it suggests that we still need to be very careful. Okay, so a little bit more about this P1 strain in Brazil. It appears also that this variant is more transmissible. It appears potentially, and again, I'm saying this is all very new, so we don't have hard data on everything yet, but potentially it could be more transmissible even than the other variants of concern. So for example, the UK variant took 3 months to become the common strain. So what we're talking about is a shift from the strain that had been dominant before to this new variant of concern being the one that is infecting most new transmission events. Okay, in Manaus, that P1 variant only took 1 month to become the dominant strain. Now, of course, there are lots of other factors that go into this, right? The fact that so many people had been infected before and things like that. But potentially, there is concern that this P1 variant could be even more transmissible than not only the original strain that was circulating there, but also other variants of concern. The P1 variant also shows mutations that may interfere with the antibody response. So similar to what I showed you with the B117 variant, this P1 variant has some mutations that specifically seem like they would interfere with antibody response. That, of course, is a concern for both reinfection or your body's defense against it, as well as vaccines. But I want to emphasize that even more than the other variants that I have talked to you about, we know very little about this variant. I looked yesterday on Google Scholar. There were only 5 papers that even mentioned this variant. And that was mostly mentions. There weren't a lot of articles that actually provided any data about it. So we're at a critical point, similar to what we saw, in March when people were starting to talk about SARS-CoV-2, where we just didn't have enough information to really understand what was going on. So I think that these are things that it's good for you to be aware of. it's good for you to keep an eye on, but also keep in mind, as you know from listening to the podcast about how to communicate science, there is a great level of uncertainty within science, and we have to be willing to acknowledge that, and that the information we understand about these variants may change, definitely will change over time. Okay, so as many of you have asked, is there evidence that the vaccines are less effective with these new strains? The short answer is it's too early to tell, but I'll try and dive into this a little bit more. So Moderna actually has a press release that's specifically related to the UK and the South African variants. And a lot of what I'm going to show you about the vaccines and these new strains is going to focus on the B117 strain from the UK, and then the variant of concern from South Africa. And that's because, as I've already said, this situation in Brazil is very new. We don't have a lot of information about it yet. And so I think, honestly, like, a lot of the vaccine companies just haven't had a chance to even check how their vaccine will work with this new variant. And, you know, I just want to say we're still getting a lot of questions that are not about the different strains and vaccine, but literally just questions about vaccines, which I've asked multiple times to hold off until the vaccine lecture. So please do that. And we'll keep talking about these different variants and potential interactions with vaccines. Okay, so what the Moderna press release says is, for the B117 variant, neutralizing antibody titers remain high and were generally consistent with neutralizing titers relative to prior variants. So for this variant that originated in the UK, the B117 variant, it looks as though the vaccine is responding similarly for this variant as with the original strain that it was developed for. No significant impact on neutralization was observed from either the full set of mutations found in the B117 variant, or from specific key mutations of concern. So those are those mutations I mentioned that could potentially be problematic with the vaccine. Although these mutations have been reported to lessen neutralization from convalescent blood and to increase infectivity, blood from the phase 1 participants and non-human primates immunized with the mRNA-1273, that's the name of the Moderna vaccine, were able to neutralize the B117 variant to the same level as prior variants. So for B117, this is looking relatively okay. It seems to be as effective with the variant out of the UK as the original strain that it was developed for. Okay, the concern becomes, however, that with that variant that came out of South Africa, which is being referred to here as B1351, they say for the B1351 variant, vaccination with the Moderna COVID-19 vaccine produces neutralizing antibody titers that remain above the neutralization titers that were shown to protect non-human primates against wild-type viral challenge. While the company expects these levels of neutralizing antibodies to be protective, pseudovirus neutralizing antibody titers were approximately 6-fold lower relative to prior variants. These lower titers, and this is the important part, these lower titers may suggest a potential risk of earlier waning of immunity to the new B1351 strains. Okay, so they're saying it still is protective, but potentially that effectiveness of the vaccine would wane earlier. So what does that mean? This is a publication from the British Medical Journal. So this is from the 26th of January. They expand on this press release from Moderna a bit more, so I want to share this with you. It says the US drug company Moderna has announced that it is developing 2 new approaches to emerging variants of COVID-19 after studies showed that its vaccine had a reduced level of neutralizing titers to the South African variant. suggesting that immunity may wane. Okay, so that's what we just heard from the press release. Although the studies showed that Moderna's current vaccine, known as mRNA-1273, was effective against both the UK and South African variants, a 6-fold reduction was seen in neutralizing titer levels to the South African variant. Okay, that's that concerning part we just heard about. Moderna says that out of an abundance of caution, it was starting a clinical program of 2 booster approaches to increase immunity to the new variants. Moderna's chief executive officer said, "We believe it is imperative to be proactive as the virus evolves." So one approach that they are taking is thinking about if they can give an additional booster. So I believe you know now that these 2 vaccines that are being distributed in the United States both require 2 shots each. And that 90 to 95% effectiveness comes after, a few weeks after you get the second shot. So what the chief executive officer of Moderna is suggesting is that rather than having the first dose, then the second booster dose, you would actually have 2 booster doses, which means 3 total shots in order to be effective against the South African strain. Now, keep in mind that this is something they are saying they are testing. They don't know that that will be an effective way to deal with this new variant, but it's something that they want to test out and see if that would actually then make their vaccine as it exists right now able to deal with this new variant. In terms of these different variants circulating, there has also been commentary about the need to really focus on safe behaviors. And in particular, the concern about the selective pressure that vaccines may be putting on the virus because of these different variants and how selective pressures may then select for one over the other. So similar to how, I talked about how humans kind of artificially selected the types of traits that we want to see in dogs, a similar thing could happen by accident with people kind of selecting more dangerous variants to circulate in the population because the vaccine then, similar to what we saw with antibiotics, the vaccine is more effective against these other variants. And then what we have, more effective against the original strain. And then what we have is these other variants becoming more common in the population. And so similar to what I said about, being careful to make sure you continue practicing safe behaviors, there was this article in the British Medical Journal from the 18th of January, in which it says, in an interview with Andrew Pollard, who leads the Oxford vaccine clinical trials, said the crucial period will be when lots of people are vaccinated, as this will put the virus under a lot of pressure. So that pressure is selective pressure, right? That's why this lecture is on mutation and selection, because we're talking about the selective pressures that the virus is under. So the virus will be under a lot of pressure. When that happens, some viruses just can't compete against that immunity. Will it mutate instead? Keep in mind that will it mutate instead is kind of missing the point. The mutations already exist, right? But then the selective pressure can cause those mutations to become more common in the population. With this coronavirus, we don't know the answer to that question yet. And that's why surveillance is going to be critical in the year ahead, to make sure that we're not in a position where at the point of population immunity, the virus escapes. And if it does, we need to know that so that we can redesign the vaccine. He added that Pfizer, Moderna, and Oxford vaccines are relatively straightforward to redesign for a new variant. And I think that last part is also really important. That one, so far, I had some good news slide. So far, it appears that the vaccines that we have are at least partially effective with the variants, the new variants of concern. But in addition to that, the technology that exists for these vaccines is able to be adjusted based on these new genetic sequences from the variants. So I want to point out a few additional things. One is that although we are referring to these new strains based on where they were first detected, the variants discussed in the lecture have been detected in the United States. The UK variant, for example, is in many different states already. Also, it is possible for new mutations to occur in the US. Remember what I said at the beginning about mutation being a random process. So, you know, it's occurring randomly. Some people have been asking questions about, like, why did it happen in the UK? Why did it happen in Brazil? Why did it happen in South Africa? And it isn't that those places necessarily were places where a mutation was more likely to occur. Mutation is random, so mutation can occur anywhere, and it is occurring probably everywhere. But also keep in mind that it is more likely for mutations to happen the more opportunity there is for the virus to replicate. The United States has given the virus a very expansive breeding ground in which to replicate. And so I think we do very much need to be concerned about the variants of concern that have already become have already started circulating in our country, of which the ones that we've been talking about have. We also need to be worried about the new mutations that could occur anywhere and then either come to the United States or have originated in the United States. So, you know, I think that this is something that we don't want to think of it just as like that's something that happened over there, and that's not a concern here. Related to that last clip that I was reading, the vaccine technology that we're using, as I said, can be adjusted. So these genetic changes are being tracked. You could see that where for each different variant, they're noting what the genetic changes are. And then they can adjust these mRNA vaccines to deal with the new genetic code for these different variants. Now, that's great. And overall, that means it's not going to take 10 years for us to try and figure out new vaccines related to this virus as it mutates. However, even that takes time, right? I mean, I think that we should all be very impressed with how efficient and quick this vaccine development has been, and how quickly it will be able to be adjusted. But even that takes time. So, you know, the development maybe takes a matter of weeks. That really isn't too bad at all. The testing that will be required, we already know that can take a matter of months. And while overall, that's, you know, from a scientific perspective, that's incredible to have a new vaccine that would be able to target these new variants more specifically in a matter of months. That's great. But as we know, as we count the number of months that we have been in this pandemic, even just that amount of time would be really difficult to deal with. So I want to reiterate again that I think it is so important that we continue to take precautions, wearing our masks, avoiding gatherings, keeping our distance from people that we're around, trying to avoid being indoors with other people as much as possible, so that we can hopefully reduce transmission rates at the same time that vaccine distribution is happening. And in combination, we could have this under control, you know, relatively quickly. It's not going to be that quickly, but relatively quickly. And so I think that if we can do our part trying to make sure that we take whatever safety precautions we can. Because, you know, similar to like you saw with the peppered moths, also like I mentioned with the mice, what we can see here in the aftermath of these different variants existing and then the selective pressure of the vaccine is that you could see a variant of concern, maybe that's more deadly, probably that's more transmissible. surviving even after people are vaccinated and then becoming the dominant one to be concerned with. As we've talked about in this class, this is not the worst case scenario for a respiratory disease outbreak. We could have a much deadlier virus, and a respiratory disease outbreak with a very deadly virus, particularly with the incubation time that SARS-CoV-2 has, that we would be seeing even worse results than we are already seeing. Not to say that we aren't seeing horrible numbers right now, we absolutely are. So, it could be worse. And I don't say that to scare everyone. I say that to encourage us all to continue to support each other, to do what is best and try and reduce transmission as much as we can. And I know that many of you in this class have suffered because of COVID-19 and have lost loved ones because of it. And, I think it's, as we feel so tired and sick of dealing with being closed in, now it's snowing outside and it's not as nice to hang out outside. I think it's good for us to remember why we are taking these precautions and the fact that people are continuing to get sick and die. I hear that from you, and I want to make sure that you know that I hear that and I feel for all of you who have been struggling with this. And it's also something that's just common and really rampant within our community. I think a week has not passed at this point where I don't hear about a friend or a colleague who has lost a loved one to COVID-19. And so this morning, for example, I woke up to news that an anthropologist who was on the brink of being a mom, so an academic, an anthropologist, and a collaborator of one of our other WashU faculty, unfortunately died of COVID. And it's things like this that make me really remember that even though this is a long and hard and difficult time, and sometimes I wish I could just give people a hug, that we are doing this for a reason, and to whatever extent we can support each other to continue to try and keep up these behaviors, to try and keep each other safe and keep our community safe, and try and connect with each other to prevent feelings of massive isolation. I think that I want to help do that for all of you, and I hope that you can all do that for each other also. So with that, I want to take some questions. I think a lot of these questions we got to in terms of talking about them, and so a lot of these questions are about vaccines. Some of them are related to these different variants. So I want to focus on the ones that are related to different variants. Some questions are about what do we know about different variants in other viruses. So in other viruses, for example, I mentioned Zika virus. That's one that I studied, right? And with Zika virus, the variant that we had in the Americas is a variant, right? So it's a different strain. Variant and strain often used interchangeably in these conversations. The Zika virus that we have in the Americas is a different strain than what was originally discovered in Uganda in the mid-1900s. And that strain is associated with particular mutations that are known to cause neurological issues. And in particular, that's why there was this issue of microcephaly in babies that were born to moms who became infected with Zika virus while they were pregnant. That was not an issue in other strains of Zika virus. So we know that this issue with mutation happens in other viruses, and obviously is something that we're aware that we need to be concerned of. Okay, so related to that, someone said mutations are common with viruses. Why are we freaking out about them? And I would say that's true. Mutations are common with viruses. Mutations are common in bacteria. And yet people, freak out when there's an antibiotic resistant bacteria because, they kill people. So yes, something being common doesn't mean it shouldn't be something that we're concerned about. And I don't know if, I think I care a lot about the language that is used, and I've mentioned this in this class before, but the idea that people are freaking out, like somehow having a disproportionately strange reaction or negative reaction to something that's just common, I don't think that's necessarily helpful. I think a lot of people have sacrificed a lot of things in order to try and improve the situation in their community during a horrendous time in our history. And I really appreciate the effort that people have put into that. I think that something being common does not mean it's not something that we should be very, very concerned about. There are lots of things that are common that we should be very concerned about. And mutations that lead to more transmissible, more deadly viruses, even viruses that cannot be controlled through the medical responses that we have developed. All of those things are reasons to be concerned. There's another question here. Let's see. So some questions are about if people have already started creating new or altered vaccines to address the new strains. And so I think, yes, I think there are a couple comments to this. One, they've already talked about, they already have the genetic sequences of the new variants. And I know that the mRNA labs that developed the vaccines have been using those to think about how they could alter that vaccine. In terms of making new vaccines, I don't know to what extent they're actually starting that process, given that right now it appears that the current vaccines may be effective, right? But as you saw from that Moderna press release, they are testing out if different dosing would be effective. And so, yes, I think people are actively working towards figuring out how to adjust the vaccine, the vaccine issue in terms of these new variants. Somebody asks if the US has the most number of cases right now, doesn't that mean the potential for mutation should be the highest here as well? Yeah, I mean, and again, we probably have seen many mutations come through the US, but remember that many mutations are neutral. They don't make any difference for the survival or reproduction of the individual that has that mutation. Many of them are deleterious so that they don't persist in the population, and only some of them are beneficial to that individual. And, it's completely random. So, luck of the draw that the US hasn't necessarily been mapped out as a place of a new variant of concern. There's another question here that says, I noticed the scholarly article for today is not peer reviewed. Therefore, how do we know that the data claims are legitimate since we're not experts in this area and don't necessarily have the academic background to check on that? So I think that's a great, good catch on that. Keep in mind that I'm sharing different types of articles with you. So I'm sharing podcasts, video clips, peer-reviewed articles, non-peer-reviewed articles, popular pieces, and so on. The one that and so you should read them all critically. The one that I shared is a preprint that's out of a platform where they are not peer-reviewed, but prior to sharing that preprint, the preprint meaning that ultimately the plan is to submit it for publication, they do screen them. So there's a screening process for that particular platform. That platform is run by a group of organizations, including Yale, including the British Medical Journal, and some others. So, it's not a completely random source, but I think it is good to read it critically. And I'll also mention that, at times like this, when people are trying to get information out as fast as possible, it's common for us to see preprints appear before they are actually peer reviewed and published. And that's because, they want people to know the information as fast as they can about that particular topic, or in this case, that variant of concern. But of course, as we've talked about, even peer-reviewed publications have their problems. So be sure to read with a critical eye. There's also a question about how are mutations of the virus going to affect the ability for us to consistently have an effective vaccine and allow us to maintain a state of normalcy? All right, this is a great question. And I want to get back then to that point that I was making about we still need to do our part. This vaccine rollout will be most effective if we continue to try and practice safe behaviors and think really carefully about the types of interactions that we're having with people. You know, you think about a place like Australia, which is basically back to a semi-normal state because they took serious precautions at the beginning. So the things that we can do to try and get back to that semi-normal state is to take this seriously and continue doing our part to what extent we can to reduce transmission and hopefully make this vaccine rollout a huge success. So these are all great questions, and I look forward to talking to you more about this stuff next week. Next week, we will have several guest speakers, including on Wednesday, someone who will be talking about the K through 12 projects. So make sure you look over that assignment and you come with your questions. I hope that you all have a wonderful weekend. I will see you next week, and until then, stay safe.